Welcome to my presentation on DNA and RNA. So what is DNA made up of? DNA is a polynucleotide made up of repeating units known as mononucleotides or a nucleotide. Each nucleotide contains a deoxyribose sugar, a phosphate, and an inorganic base. Examples of the bases found in DNA are adenine, thymine, cytosine, and guanine. The base guanine always binds with cytosine in a process known as complementary base pairing. 3 hydrogen bonds will form between guanine and cytosine. The base adenine and thymine will bind together as well in complementary base pairing and will have only 2 hydrogen bonds between each of the 2 bases. DNA is arranged tightly in a coil structure known as double helix and is associated with proteins known as histones. DNA is a double-stranded molecule. So here's an example of the picture of the DNA molecule. We can see that between A and T, we only have 2 hydrogen bonds, and between C and G, we have 3 hydrogen bonds. The bonds between the deoxyribose sugar and the phosphate group are known as the phosphodiester bonds. What is the function of DNA? DNA is an incredibly stable structure, making it able to be passed on from generation to generation with very few mutations, due to the fact that it has a phosphate sugar backbone, which helps to protect the chemically reactive inorganic bases, preventing corruption from, let's say, chemicals or other agents. DNA also has weak hydrogen bonds between its 2 strands, allowing for semiconservative replication to occur more easily, or even protein synthesis, as both these processes require the strands to be able to separate. DNA also is an extremely large molecule and therefore can store a lot of information in a very small space due to the way that it's tightly coiled around histones to form a double helix. So the process of DNA replication is known as semiconservative replication, and this takes place in S1 of interphase. In the first stage of semiconservative replication, DNA helicase will cause the hydrogen bonds between the 2 strands of DNA molecule to break, thus causing the strands to separate. The original DNA strands will then act as a template to which 3 nucleotides will bind to in a process known as complementary base pairing. Next, the DNA polymerase will synthesize the phosphodiester bonds on the new strands. As a result, two molecules of DNA are produced, with each molecule containing 50% of the original DNA and 50% of the new DNA, which is why the replication process is known as semiconservative DNA replication. The evidence that supports the semiconservative replication of DNA was found by 2 scientists known as Messelin and Stahl. Their experiment basically works on 3 key principles. The first, that bases always contain nitrogen, and that the fact that nitrogen-14, which is an isotope of nitrogen, is lighter than nitrogen-15, and that bacteria will always use the nitrogen from the growth medium that they are exposed to in order to make the DNA. So in the experiment, what happened is that the bacteria were grown in nitrogen-15 media and allowed to grow for 1 generation. They basically then centrifuge the bacteria to actually obtain the DNA and to work out the density. Now, we can see that in the test tube in the diagram that the density is much higher than that of the density of nitrogen-14. Now, then what happened is that they moved the bacteria from a nitrogen-15 medium to a nitrogen-14 medium for 1 generation. This is quite significant because it means that the results suggest that the density lies between nitrogen-14 and the nitrogen-15, therefore suggesting that the semiconservative replication must have taken place because the DNA contains 50% of the old DNA and 50% of the new DNA. The DNA is like a mix of the densities, therefore causing it to come out in the middle. So what are RNA and what are the key properties of it? So RNA is a single-stranded nucleotide. It's made up of the bases adenine, uracil, guanine, and cytosine. The base that's been substituted has been uracil for thymine. It also uses the base ribose for the sugar rather than deoxyribose. We'll go into RNA in more detail later in another presentation. If you like this, please subscribe. Thank you for watching.